All right. And we're good to go.
All right, should I get started?
Yeah, whenever you're ready.
All right. Can we see my screen okay?
Yep.
Cool. So as you all know, I am Dr. Geoff Dilly. I am a biology professor here at Channel Islands, now an associate professor as of this year. So I got tenure, which is great. So you're stuck with me now. And I want to talk to you mostly about what I do here at the Channel Islands, but I am gonna, because it's cool, talk about what I did as a graduate student working here. So this is actually a picture I took outside of Alvin, the submarine. And what you can see there, that's actually a high pressure mass spectrometer. So my PI actually built a mass spec that can operate at high pressure and sniff a vent, so you can look at the chemical profiles of a vent, similar to the the GCMS that you might have in a chemistry lab, although quite a bit more hardcore. But I've gone from this environment to this environment, which is, as you probably recognize, Santa Rosa. And I wanted to talk today about what I do in the field, which is where I'm going to focus. I had a fair amount of slides for what I do in the lab, but I cut them for time because I think I'm assuming you guys are much more interested in the field stuff because it is all on Channel Islands. And then I have some fun pictures at the end. So I consider my full title of my position, I would say, is a marine ecophysiology professor. And so I wanted to sort of share a secret that marine biologists learn, which is that you can take the word marine and you can put it in front of any science and it still makes sense, because all sciences can exist on the majority of our planet, which is marine systems. 98% of our habitable realm is the marine system because we have 3 dimensions to play with, not 2. I guess birds can play in the 3rd dimension, but they're minority. But there's also very, very interesting things that go on with the geology and the chemistry and the physics. Although marine physics, we call it, it's catchier to call that oceanography. And even within marine biology, there are subdivided fields, and I know people who are in all of these. So you can be a marine microbiologist. You can be a marine virologist, right? There are people that study marine viruses. You can be a marine ecologist and more. So if you have a term in science, you can basically put marine and you can study those systems in a marine environment rather than terrestrial. So what do I mean by ecophysiology? It is a portmanteau of ecological physiology, which is sort of the intersection of how an organism interacts with its environment and how it will respond to stress. Stress is, you can all feel it. What you feel as stress for exams is both a mental situation, but it actually manifests in physiological conditions, right? You feel like you get sick because of stress, because your body spends a lot of energy dealing with your mental taxing access, that your immune systems can actually be depressed by severe trauma or stress. But what I mean by stress really are forces that are constraining an organism due to tension. So a stress is usually some sort of factor, either biological or abiological, that alters an existing equilibrium. The easiest one to imagine is temperature, right? You are very stressed when my wife's heading to the hospital. She works overnights at the pediatric ward. You can understand that equilibrium can be disturbed if you exit what is your nominal conditions. So I like to use my model organism, which is Fry from Futurama. And he is basically put through some stressful influence during every episode, and that gets him to disequilibrium. But what's really important to think about when you think about stress, equilibrium, and physiology is that if you can acclimate, you don't go back to where you were. You go to a new equilibrium, right? Because your organism has acclimated. You may change your proteins, right? My background is actually in molecular physiology. So I looked at protein adaptations and changes at the molecular level. You could be doing ecological changes. You could have behavioral changes. All of these are ways that you could acclimate to your situation. That's not the same as evolution, right? That's adaptation. That's generational. We're talking about what an organism can do to modify its own response to an environment. And stresses come in a few different flavors, right? So we've got biotic ones, things like predation or competition, both of which are intensely important in the intertidal zone where I do my research now. So much so that when you go to the intertidal zone, you can see these bands of organisms. Basically, those are defined by predatory bands from below or competition for space. It's really high premium for space in the intertidal. Or they're driven also by thermal stresses, acid-base stresses, oxidative stress, desiccation, toxins, right? These are all abiotic conditions that can cause stress to an organism. And there are, we call it the 4A's in physiology, the responses. You can have adaptation, the evolutionary response. You can have avoidance. If you can get out of there, you will. You can have acclimation, or you can have acquiescence, which is a nice way of saying you can die. And so to look at how these things are responding, you need a few things. So what I sort of always look for, and when I'm grading papers, I always look for this when people are writing their proposals. You need to pick an organism that is appropriate for your question. Not every organism is a good test subject for every question. Can you raise it in a lab? Do you need to raise it in a lab? Is it amenable to field studies? Does it vary on the axis of stress that you're interested in? Does it have a good comparative? Do you know the genetics of it? Do you need to, right? Is there a population base? These are all questions you can ask about your organism, and different organisms seem to have fits for different questions. One of my sort of heroes is the late, well, very late, he's like 100 years dead, August Crowe, who won the Nobel Prize in 1914 for doing the first physiology experiments on goldfish. And before you go, goldfish, they are some of the toughest organisms on the planet. They can survive practically anoxic conditions by switching to air breathing, which is amazing. Almost no fish can do that, but they can gulp air and then force it through their gills and saturate that. They can accept a temperature range from 0 degrees Celsius to 40 degrees Celsius. And remember, they're ectotherms. That means their body temperature can change by 40 degrees Celsius. So he studied their physiological responses, and he had a really cool quote that I'll paraphrase it here. For every question, there exists out there an animal to answer it. And so it is up to you as a scientist to understand what animal, what organism, what plant maybe answers your question most appropriately. You also need a dynamic environment. It does not work to study, say, temperature stress in the Antarctic unless you're looking at, say, climate change, because the Antarctic is incredibly stable. They're so stenothermic at that point that your temperature ranges for the marine environment there is minus 1.7 to minus 1.3 Celsius. That's it. Your entire range is less than a degree Fahrenheit. So you need to pick an environment that is dynamic. Now, if your interest in the environment of Antarctica is brining, right, because it becomes more or less briny based on sea ice, or if it's looking at long-term changes like ocean acidification, then maybe that is the correct dynamic environment. You need a system to isolate your variables. If you're doing all of this work in the field, then you're not really doing physiology. You're just really doing ecology. So you need to be able to simulate some of the things that you're seeing in the environment and bring it back to the lab. Now, I'm not a true physiologist. I like to go to the field and I like to do work out there. Most of what I'm going to talk about today is the field side of it, but know that I spend just as much time designing and replicating those conditions and trying to isolate the variable that's of most interest. So if temperature is what I want to study, I want to hold everything else as stable as possible so that temperature becomes the defining aspect for my question. And finally, you need a good question. You always need a good question in biology. That's going to be the decider of whether you have a cool experiment or not. And so I want to go through a couple things that I've done in the past. My PhD, I looked at a group of worms called Paralvinella. They are related to the Pompeii worm, which you might have heard of, which are some of the highest heat tolerance animals on the planet. This little guy here, about half the length of your thumb, it can survive 55, 60 degrees Celsius almost permanently. Now, at that temperature, proteins are starting to break apart. And so a lot of what I did at this level was actually molecular because we couldn't quite understand how their proteins were able to be stable at this incredibly elevated temperature. They live very close to the vents themselves. They're the closest of all known species that live to the vent effluent. And as you approach a hydrothermal vent, you see there's a lot of similarities to the intertidal zone because although it is crushing pressure depth, right? And it's also exceptionally cold, 3-4 degrees Celsius around, you have this really interesting geospatial thermal gradient between what is the chimney spewing out 400 Celsius and ambient temperature. And in that gradient, you see a temperature range that goes all the way from 4C up to about 70C, where animals are habitating in there. Above that, you get archaea and bacteria to about 130C and then nothing, right? Just chemicals. So what I did was I actually brought these animals up aboard the ship. We use this thing called the slurper, which does exactly that. Puts it into a chamber. We bring it up. I sorted the worms out. That was a tray of them. A couple species that were intermixed there. I put them in these, essentially they're gas cylinders, but I filled them with seawater, and I pressurized them using HPLC pumps. So I cannibalized an HPLC pump, and instead of running oil or fresh water through it, I actually made them run seawater, which meant they decayed pretty quickly. I learned a lot of repair on that. And then I put them into heat baths. And that allowed me to look at the thermal tolerance of them at a lot of different conditions. In my postdoc, after I got my doctorate, I went to UC Santa Barbara. And with that group, half of my time I actually went to Antarctica. And I studied down in McMurdo Station here. And this is a setup, although I was involved in this research, I didn't set these tanks up here. I did this in Santa Barbara, but by the time I got there, somebody else had set them up already. But this is essentially, the blue is low CO2, the yellow is medium CO2, and the red is high CO2. And this was basically growing up the larvae. There's a 4-cell stage of the larvae growing up from the Sterechinus neumayeri, the Antarctic sea urchin. It was growing under these conditions. And this was a question for the future. How will these organisms evolve to respond to these molecular stresses of growing up in an acidifying environment? How is it going to affect their formation of their test, their shell? How is it going to affect their spine formation, their fertility, all of these particular questions? So I did a transcriptomics research there, a lot of sort of gene expression analysis. And now, in the last 6 years, I've shifted my gaze a lot shallower to the intertidal zone. Here's a number of different students from the years, all the way from my first couple students. That's Ryan Newkirk from biology and Paul Spar from ESRM. Paul Spar founded your drone program, by the way. I actually founded it with him. It was my first semester here. Anyway, so we were working out on the intertidal zone. This is Bechers Bay. This is Bechers Bay. This is actually Skunk Point, but we'll show you those later. And it is in conjunction with a lot of different research groups and larger organizations that are interested in this. And my overriding question here are, how are environmental shifts affecting the intertidal population in the Channel Islands? And I keep that purposely vague because there are a lot of different elements that I'm taking out of this. Just a little bit of background for you. These are the 20 of them are active sites that the NPS currently monitors. And you can see there's actually 5 on Santa Rosa, as well as, 6 on Santa Cruz, 3 on Anacapa, 4 on Miguel, and a couple on Santa Barbara. I have gone to do sampling at a number of these. I have not yet been to Santa Barbara or Miguel, actually, so I still have to get those 2 islands, but I've gone to help them at different sites. But you'll note that there's a big space here on the north side of Santa Rosa. And so that's where we came in. So what the NPS does, and this has been going on for a long time, since 1982, first permanent ones in Anacapa were set up, is they have these fixed plots. And you can see you'll see, and if you've been to, has anybody here been to Bechers Bay? Have you seen my bolts?
I have not actually.
Okay, well, if you ever go out to get a chance to go out to Rosa, Bechers Bay is right next to the station, and you'll see a bunch of bolts like this. Careful where you step when you see them, because those are my plots. But what you have here is these bolts will be laid down, and what NPS has been monitoring, they set up these fixed plots near where the barnacles are, thalamus and balanus, turfweed, which is known as endocladia, or it's also called wire brush. It's like that grainy brown stuff you've seen out there, looks like that. Rockweed, which you've definitely seen if you've ever been to any intertidal zone around here, that's Sylvidia. And then mussels, which is what I tend to study, which is Middleus californianus here. And they have these grids where they just count 100 plot points. So you have 10 lines this way, and you put a bar that goes across, and you just count what organism is under there 100 times. So there's sort of a micro high resolution analysis of what's growing here. The nice thing is it's long-term, right? This is long-term ecological monitoring. And what is true of all LTERs, or long-term ecological monitoring sites, research sites, is that the question isn't answered in a single trip, right? In fact, you may not even have a question when you go out there. It's more about establishing baselines and building out understanding of what is there and how does that shift over years. So to that end, actually with the late great Cause Hannah, on the day that we founded the station, actually the inauguration, right after everybody left for the party, he and I took a hike out to Bechers, and then with his family out to Skunk, and sort of identified a couple places where we could set down long-term monitoring sites. So that's where we have 2 new sites that have been monitored since, I guess, December 2014 or maybe January 2015, somewhere around there. And at these sites, I do 4 main protocols. We're adding a new one in this year. I haven't put it in yet, but we're adding in some algal monitoring as well with a new student. But the 4 that I've been doing for a while, Photoplots, doing the same thing that the NPS did, counting organisms in a grid. The next one are vertical transects, where we lay out a transect line, and we actually use the entire site as a grid. We count the organisms on that. So that gives us a broader but sort of lower resolution image. Think of it like taking one blurry panorama of the entire Grand Canyon and then a couple close-ups here and there where you get some high resolution imagery. So that's the kind of concept. And speaking of imagery, we actually do some of that out on there. We take Phototransects, which I call swath assays. So instead of taking a point, you take a swath. So we have a meter by meter grid, and I have a camera that we've built in with this PVC frame. It's a little unwieldy, but we have a Bluetooth camera, and we take it and we march the camera down the transect lines, and then we use software to stitch these together. The goal of this eventually is to have machine learning be able to tell us what percentage of each of these sites are actually covered by different organisms. So that's the goal. Again, working with computer science on that. And the last one is getting some information about the abiotic conditions out on the island. So we've placed a bunch of these little eye buttons, which are like thumbnail-sized watch battery things that just measure temperature. They're pretty dumb. They don't have a lot of resolution. They can only really resolve to about the nearest degree Celsius. They say ½ degree Celsius. I wouldn't trust it. But a degree Celsius is fine if you're talking over years, which is what we're doing. We have a button at each one of our plots, and we have 20 plots per site. And each of these plots are co-locating temperature with the organisms that we find. So I can co-locate the abiotic with the biological. So here's a picture of the vertical transects put out there. We have a bolt at our 0 plot, and then we have another bolt at 30 meters. So both sites are 30 meters wide. And then every 3 meters, we step down a 25-meter perpendicular transect line like this. And we count at every 25 centimeters. So do the simplest math, 25 centimeters, a centimeter is obviously a 100th of a meter. So 25 centimeters, 25 meters, that means I get 100 points for every meter over a 25-meter line, and we have 11 transects. And yes, that works because we have a 0 line and a 30 line, so 0, 3, etc. So we have 11 transects, we have 100 points, we have 1,100 data points from each transect that we go out to do, and we do this a couple times a year at each site. And that gives us some basic positional species data of where you're finding things in different zones. And then the Phototransects, this is kind of what it looks like. They're pretty high resolution images. We're using a 12 megapixel Canon camera that I bought for this. And it gets stitched together. We take a 65% overlapping photo. Technically, it's only 35% overlapping on each image, but because there's multiple images, it ends up overlapping at 65%. So this is a 1 meter by 20 meter transect. We use this thing called Image Composite Editor, which is a free program by Microsoft that will take each of those images and then overlay them and stitch them together. So this is what it looks like. I've hopped up on a couple cliffs to take a picture of this. This is what Bechers Bay looks like. There's our 0 bolt. There's our 30 bolt. And you can see that we've laid out 11 transect lines going out 25 meters at each. We do the same thing at Skunk. It's a little hard to see because the green sort of blends there. But what you can immediately notice, even with an untrained eye, is the differences between zones, right? So we have 4 main zones in the intertidal zone. This is called the low zone. And obviously, we only go out during very low tides. So these are minus tides. This area is mostly underwater. So most of the day, in fact, sometimes our thermaloggers, speaking of computational biology, it's very cool to watch their graph because you'll see that they stay underwater for many days, and then you'll see these big spikes because it goes out of water, and suddenly it's either the middle of the day and it's hot, or it's the middle of the night and it's cold. So think about a wave of temperature for daytime, nighttime, daytime, nighttime, and then overlay a water temperature, which only really changes about a ½ degree Celsius per day because the ocean doesn't really change that much temperature. So overlay the two of them because it switches from one wave to the other when it goes out of water or when it goes back into water. So the low zone ones spend most of their time telling me what temperature the water was, and then they get these pops. But remember that it's an interesting graph, but remember that the organisms that live in each one of these plots, that's what they're seeing. So they have these very unique, very interesting profile lives at the low zone, the mid zone, at the high zone, and then way up at the splash zone. The splash zone almost, in fact, the splash zone is never inundated. Sometimes it gets hit, right? So you do see some barnacles up there, but there's almost no actual water getting up here except on exceptionally high king tides like we had a few weeks ago. So I wanted to take a time here. So if everybody's paying attention right now, this is the most fun slide of my talk, as long as my computer doesn't crash. So what you're going to see this is the stitched image, and we're going to walk through the tide zone. On the left is the Phototransects stitched for Bechers, and on the right, it's stitched for Skunk. And you're going to see similarities and differences, because even though these sites are only 5 miles away, they actually do have some unique geomorphologies that lead to biologies being different. So there's really sort of micro environments here. So here we go. So that's 0 meters. Now we're at 5 meters. We're at 10 meters, lots of rockweed there. Anemones over here, more rockweed. There's the mussels. There's Phragmatopoma, more mussels. So that's it. So I'm going to play that one more time. So let's go through one more time to see Skunk Point and Bechers Bay. All right, so just as a side note, I played this for a talk I gave at Pomona a couple years ago, and it melted their MacBook Air that they gave me because that was like a gig. So fortunately, I'm running on my own computer right now. But we did these you can see here are all of the transects, just sort of zoomed out. There's Bechers Bay and Skunk Point. And you can see we walked a little bit of an angle here on this line. But more or less, this took a lot of work. And we have these really pretty swath assays, of which we're really hoping to be able to automate, right? There's so much data in here that if we can, and it's so much higher resolution than if I just flew a drone. Like if I flew a drone over there, I'd never get nearly as much information, and with that high resolution. I mean, that I'm not kidding when I say each one of these pictures is about 10 gigabytes of information. And so with that much information, if we can start to match pixels, if we can start to match shapes, if we can use machine learning, then we can build algorithms that tell you, what does an anemone look like? Okay, then how many are here? What does rockweed look like? Okay, how much does coverage exist? And then the questions become interesting because you can look at, say, what's an annual shift if we do this winter and summer? What is a decadal shift if I start looking at information from 2015 versus 2025, for example? This is what it looks like if you just sort of overlay that on top of the USGS map of the island. That is exactly where those lines are. In fact, the rocks even line up perfectly. So we have geotags on these, so they actually line up pretty well with the imagery. And then that's what it looks like. There is the bunkhouse, if you've been there. You guys have all been to the bunkhouse? Not everybody? Oh man, you guys, shameful. You got to get out to the island.
I've tried like 3 times,
Oh,
and then
sorry.
COVID and stuff.
Yeah, it's been really hard for us to get out there. We had to sign so many forms just to get a small group of 4 people to go do our long-term data set. Like they're just
The plague. The plague, I don't think many of us have been anywhere.
Yeah, no, this area is about all I've sat in for months. But you can see that's actually the boats there that day. So it's really close to the station, and so make it a point to get out there, even if it's after school's over. Just go to Santa Rosa and visit. You can see my site. It's only about a quarter mile from where the boat docks. Skunk is a bit more of a commitment. It's about 6 miles away. This is what it looks like zoomed in and then zoomed out. And so this is on this sort of rocky outcrop between this small sandy beach and this really big sandy beach, which is sort of the sandy part of it. This is actually what's known as Skunk Point, though. I always thought it was the beach, but it's actually this mountain right here that's called Skunk Point Mountain. It's short. And then the final bit that we do is the thermochrons. These are the little eye buttons here. We've logged data every month from April 2016 to present. And then what we've done, so this is just some data, some preliminary data I'm going to show you, giving you the median and the quartile temperatures presented in 2 different ways. First, I'm going to show you temperature per month at each site and per zone. And then I'm going to show you temperatures experienced by the dominant species that we tend to find by quartile and median. So here we are at Skunk Point and Bechers Bay. This is 8 degrees up to 36 degrees. So really big range. And you can see that the splash zone, as you might expect, experiences the broadest range because it's mostly daytime. And it can get pretty hot just bearing down in the sun. The low zone never gets that warm because it just doesn't stay out of water very much. So this is Bechers and Skunk in April and then in May. And you note that May is actually cooler than April because of the fog layer, because you get this sort of May gray. April is a sunnier month, and so it actually gives these lower highs, even if the median is inching its way up because the water is getting a slight bit warmer. June comes back because you have a lot of brighter days in June, and the water temperature is going up. You see some temperatures way up at 35. But you see the low zone is really constricted. The reason for that actually was that in this low zone, there weren't that many really low tides. And so the low zone spent very little time out of water. This is July, and then August, still pretty low, and then September. And then I wanted to show you by species temperature ranges. So Phylospatix is a seagrass. We don't tend to see any of that at Skunk, so I don't have it listed. We do have a decent amount of it at Bechers, so you can see their range here. Middleus is mostly in the mid zone at both sites, Endocladia, Sylvicia, and then barnacles. And so you can see the barnacles have to deal with a lot of temperature stress, right? They live really high up in there. So they're seeing ranges down to 8 and way up to about 35 degrees. And then you can see that was the spring, this is the summer. The ranges get even higher, especially at Bechers Bay. The sun hits a little bit more direct. So that's pretty much the field run. I just have a little bit here about the lab work. Most of my work here in the lab actually revolves around mussels. So I collect mussels. I don't go out to the islands to collect mussels. There's no reason to go. They're the same animals. So I just go to the pier, just scrape them off the pier. I have been told by several lifeguards not to eat those mussels because of red tide, and I have to explain that I know that and I'm taking them for research, and then they usually let me go on my way. Interesting to note, there is no permit that you need to take invertebrates. You do need them for fish, right? You need a fishing license, but to take invertebrates, you can only, the only limit is that you can only take 10 pounds of mussels per day per person. So that's a lot of mussels. You know, even one person can pretty much bring me an entire research season's worth of stuff in a day. So we collect them. We put them, so you see this is the back of my larger tank in my lab here in Modoc. And we usually let them acclimate for about a month to our condition sets. We have cycling tanks, we've got filters and so on, keep the water as clean as possible. Then we usually do things like temperature experiments. Our most recent the thing we've been working on for a while, although COVID threw a lot of wrenches into that set of experiments, is microplastics and toxins. So my new stresses are how do they deal with filter feeding microplastics, and how do they deal with benzo alpha pyrene, which is an organotoxin that's found in cigarette smoke, that's found in gas, that's found in oil, that's found in car exhaust, that is in somewhat elevated areas around here. So one of the problems with BAP, or benzo alpha pyrene, is that it is hydrophobic. So it hates water, even though it can be found in the ocean. So the first thing it gloms onto are microplastics, which are also hydrophobic. So they bind to it. So one of the problems that microplastics have is that they are vectors for this. The microplastics themselves, they're not great, but they're probably not lethal by themselves to organisms. I wouldn't eat mussels that are microplastic contaminated because you are accelerating and accumulating your own intake, and we don't have a way to shed plastic. So that's not a great thing up the trophic chain. But at the filter feeding level, we don't expect microplastics on their own to be very toxic. There's not a lot of literature to support that. In fact, a lot of the literature say plastics are not a problem, which of course is itself a problem because that's not true. But one of the issues, of course, is that they can vectorize this. So to study that kind of analysis, we really want to do physiology experiments. And for me, the best proxy for metabolic change is respirometric change. So we built these little CI respirometry chambers. They're just like the ones I built at Harvard, but on a CI budget with acrylic and rubber rather than titanium and stainless steel and high level ruthenium. And then we built, this is basically a little Surface Pro. And then this spaghetti here is an Arduino board that we just basically pegged in a bunch of stirring motors. So this motor can stir the cylinder in here without having to do water exchange. So I can close the chamber off, continue to circulate it, and then take their oxygen respirometry. And we can also do genetic analysis on the back end. So we can take the tissue we do for RNA extractions, and we do analysis of their genes and their proteins from that. So that's all the lab stuff I'm going to talk about. Just sort of talking about my future directions here. One of the things that we're looking at doing right now is putting an exosonde. Aspen and Joe have actually the exosonde that I purchased several years ago. They didn't even know that I purchased it. That's how long this thing's been away. It was before either of them got hired. But I purchased this multi-parameter exosonde that I'm going to try and get permitted to deploy onto our pier. I want to, that multi-parameter can do dissolved oxygen, dissolved organic carbon, temperature, CTD, chlorophyll, all these other things to let us characterize the marine chemistry. One of the things I don't really have going right now. And then on top of that, we're going to be doing some algal analysis, looking at the different types of prominent algae that we can find out there, diatoms, dinoflagellates, both morphologically and genetically, to see if we can characterize what are the mussels actually eating out there, because that's what they filter feed. Additionally, with the computer science department, again, COVID wrenches all over the place, but the goal is to do machine learning and then continue our physiological experiments on immersion, desiccation, microplastics, toxins on mussels and other local species. All right, so you've heard all the boring stuff. Let's look at some fun pictures. So this is from an older picture from my work. I think this picture, oh yeah, so this was a picture, this is actually a Photoshop job that I did of two pictures, one of the sub and one of the ship. I was actually on a little pontoon boat here. They picked us up in Guaymas, Mexico, because we were studying vents in Baja, California. So I was on a little pier riding this little taxi boat at 6 in the morning. There was like water in the boat, and I was concerned I was going to sink on the way there. And then this is what the Alvin, the submarine, looks like. This is a pretty day on the front of the bow of the Atlantis. And that's what it normally looks like when you're plowing through the North Pacific, gigantic waves. This is Alvin, the submarine. That's its slurper on the front. They always keep these boxing gloves to protect the manipulator arms, although it's pretty funny. And then here you can see, this is what it kind of looks like when it comes back to the surface. The swimmer will jump in the water, and he is literally on the phone. It's a hard line that goes through the wall of the sub, so that if all electric is dead, if the sub just died, they still have a hard line, literally 2 cans in a string. I'm not kidding you, that's exactly what that is, so that you can talk to the pilot on the inside, explain to them what's going on. So if all cameras are dead, this guy can talk. Even without that, it's still useful because they basically turn off all their sensory equipment when they get back to the surface. It's too disorienting. So they let the ship guide it back in. This was on one of our dives. An octopus decided to, I don't know if that's attacking or loving the manipulator claw, but it definitely took an interest in it and was hanging on to it for a while. Unfortunately, we had a cephalopod biologist on board, so they snagged it. So that became a study object. That was me long time ago, many years ago, inside the sub. You can see there's a lot of switches that you shouldn't touch or things will go wrong. But yeah, the windows in there are about this big, and you and another scientist, they've redesigned it. It's actually, they built a new sub in 2016. So now you sort of lie straight forward, like on a bunk. But it used to be that you and a scientist would cross legs behind the pilot, who would sort of sit on a little stool in front of you. So the 3 of you would be stuck in there for 11, 12 hours. And then this was a picture, I think I took that through the window. They were taking the temperature of a vent. So that's a titanium temperature probe there that can go to 700 Celsius. You know, it had to be titanium because that water temperature can be 450. This was cool. This was photographed. So this is actually a camera that took an image once an hour for 2 weeks. So kind of what we were doing, but we dropped a camera down there just to sort of take a look at what a vent looked like. And finally, anytime you get on one of these on board one of these ships, you have to get baptized, because hopefully, very hopefully, you didn't get wet during this or something really went wrong with the sub. So when you come back up, they hit you with buckets of water. And if they really like you, they put those buckets of water in the freezer first and get it down to -1 Celsius, which is what we did for our undergrad friend here. You can't even see her, but she is somewhere under the bucket. That was a postdoc in my lab. I think there's another postdoc here. That's me hitting her square in the face. Don't worry, they hit me too. So only on your first dive. So you get baptized after you come back from Alvin. And so I wanted to end the talk with jobs that you could do if you wanted to be a marine biologist. And so how much school would you need? So with the amount that you have right now, you could go into being a lab technician in a marine lab. You could be an aquarist at a zoo or an aquarium, educational specialist outreach to K-12. You could be a science writer, congressional aide. That's a really cool path, especially for congressional people that believe in science. That would be a good one. But you can help them craft legislation and be a sort of interface between the public and policymakers. You could be in a field ecology assistant. If you went on and got a master's at this level, you could be a National Park Service biologist. You could be a field ecologist, go out and do those surveys, lead teams. You could be an ichthyologist, marine mammologist. You could be going out to Antarctica or to Hawaii or whatever, doing mammal surveys. You could be an environmental scientist. You can be a conservation scientist, USGS, Department of Energy, all of these other big government groups or private groups. You could be a research associate at a marine lab or go into fisheries management. And if you went to as far as a doctorate, you could be a professor. I don't recommend it. Okay. You could go into director's level research at National Marine Fisheries or NPS or something else. If you went the biotech route, even marine biotech, you could be a manager of a research team there. You could now be the curator of an aquarium. You could basically run everything. You could be a leader of a conservation team or group or even a program, or the chief researcher at a private company or a public institution. So with that, I just want to say thanks to Steve Whitaker from NPS, Russ Bradley, our current station director, Cause Hanna, our late station director, NPS Island Packers, and all of the current and former members of my Tide Fools lab team. So there's one iteration of the crew from a couple years ago. I'll take any questions.
Awesome. Thank you so much, Jeff. That was amazing. That was so cool. Like I feel like you packed so much into that, and like honestly, with the lab section, I could have heard you like go on longer.
No,
so,
well,
but it's all good. You fit
it's evening. I know people want to get dinner. I have a whole different talk for that one.
It's all fascinating. Like and it's so funny that it sounds like the most CI thing to have like that that unit that you bought like years ago, and then like kind of it got forgotten about, and then like was found, and everyone's like, oh hey, we have this here, did anyone know? Like that sounds like the most Channel Islands.
It's not cheap either. This is a $25,000 instrument that I bought with money from the institute, from the alumni association. Like it was a big deal. It went all the way to the president, and then it sat in AJ Biersad's lab, the computer scientist guy, and then he was like, I don't want this, so he gave it to Aspen. So Aspen had no idea where it came from.
That's so funny.
But finally, it's going to get out there onto our island.
Sweet. And I did realize that, so I've been to Bechers Bay, but I haven't been to that spot where the transects are. So that's why I hadn't seen the little like rods in the ground, because I've been to the island twice, but I usually like to go on the hikes that went inland. Like I've done Lobo once all the way through, and one time like halfway, and then went back.
Interestingly, we actually debated putting a site at the intertidal zone that's actually at the end of Lobo, but we decided against it because of how difficult that would be to get to multiple times a year.
Yeah, I can only imagine.
It's a long hike.
Yeah, it is.
We have to service every 2 months, and we have to take a fair amount of equipment when we do. So most of my team is taking 40 pounds on their pack, which is already a long enough hike out to Skunk and back. That's 10 miles. Yeah. So Lobo would be like a 14-mile round trip. That's a bit much.
Definitely.
Do you guys get a lot of marine debris at your locations?
Yeah, especially, well, no, actually both sites. Not a lot in my site itself because it's rocky intertidal. That tends to scour it a lot. I've seen, you know what I tend to see is traps. A lot of lobster pots that wash up, usually collapsed, but I've seen wreckage of that. I've got to move them out of there. Skunk, so I've done the marine debris protocol a lot out with them. I'm not involved in the current grant, but I was involved in the last one. And we did, one of our survey points was Skunk, but we did the sandy beach protocol, not the rocky. So we went past that, and Skunk is just littered. It's interesting because it's the center of the current that comes through between it's the channel current that goes between Rosa and Cruz. And so all of these buoys and pots and parts of ships, dead animals like dead elephant seals, parts of dead whales. I've seen dead dolphins there. Sorry, guys. Yeah, so lots of stuff washes up at Skunk because it is this like sandy spit there. I will say the saddest thing to me, corpses don't really bother me, but I've done a lot of necropsy before. But the thing that really bothered me were the birds that I saw that were filled with plastic, so many of them. Like almost all the skeletons have lighters in them and bottle caps and stuff. They think it's food, they dive down, and that's usually what kills them because they starve.
Oh man, yeah, no,
Cheery,
that is
I
very
know.
depressing. Yeah,
Yeah,
that's
don't take
something
my
that,
capstone. It's all that kind of thing.
yeah, we touched on that like briefly in Allison Alvarado's ornithology class. I took that just last spring, and like that was like a low point for sure. Everyone was like, "Oh damn!" Like,
Yeah, seabirds are in real trouble.
mhm.
I think of all so corals are basically on a clock. They're gone. At least tropical corals will not exist in the same way that we see them. Even now, they're already gone. I mean, there's only half of the Great Barrier Reef left. Probably by 2050, there really won't be any coral of large numbers. I think second to that is probably seabird populations. They're probably the second most at risk.
Are the seabirds basically in the same situation as the coral? Is
Well,
there
so there's
nothing we can do?
that's a good question. Coral is probably nothing we can do, unfortunately. I mean, yes, there are things we can do, but they should have been done in 1980. I think it's more like we're hitting the brakes too late. Even if we stop now, we're going to hit that wall with corals. But because just very briefly, the issue that corals face, it's a double hit. Their zooxanthellae, which are their algal symbionts, are pretty temperature sensitive. The corals actually aren't temperature sensitive. That's a common misconception. It's the symbiont that can't take it. And so the symbiont says, I'm out of there when it's like 33 Celsius, which it used to only hit once in a while. Now it hits every single summer for longer and longer. And when it hits that, the coral bleaches. And the bleaching of a coral is actually the symbiont leaving. That's the symbiont's the coloring of the coral. And so the corals are still alive at that point, but I liken it to like a two income household. If you're barely getting by with 2 paychecks and then one partner leaves, like the other partner can maybe hang on for a little while, but eventually they can't provide enough food for their family, right? So that's kind of the problem here, is that a coral both feeds with its tentacles and photosynthesizes through its symbiont, and it cannot get enough energy by just feeding. And so it can usually live 2 to 3 weeks if it's been bleached out, but not much longer than that. So that's hit number one. And hit number two is that corals very famously have a calcium carbonate exoskeleton, right? They deposit this hard part to protect themselves. And that's chalk, and that requires a basic environment to be able to deposit. And
Ocean acidification.
Yeah, exactly. So the mineral that they make is called aragonite, which is just a chemical structure of calcium carbonate. And aragonite has a saturation state that more or less gets passed at 450 parts per million CO2, which we are fast approaching. And once we go below its saturation state, the animal will not be able to lay down its calcium carbonate anymore. It's already having trouble. The closer we get, the more energy it has to spend to build this skeleton. And then once it gets past that, it just won't be able to change the chemistry enough to be able to make its own skeleton, which means it can't make its skeleton and it can't feed anymore. So that's pretty much a death sentence for it. The birds, it's a different problem, right? It's plastics, and it's also overfishing. That's also a problem. Both of these are eminently more solvable than, I would say, climate change and acidification. So I think with dramatic, specific interventions, seabirds could be saved, but it would require real lobbying, real policy, real action to do so, and not just sort of placating things like the ocean cleanup project with some pretty boy and slot going, I've solved climate change because I have a floating tanker and it's going to go pick up a bit of trash. Like, that's not going to clean the entire ocean. It really requires dramatic change of stopping using consumable plastics. I think that's possible. I leave the sociology to other people, but I think that's a possible solution.
Really quick, to ask, just because, well, kind of separating from like the sadness of the birds, how cool was it to take a picture next to those penguins?
Very cool. Very cool. So you can approach them, right? They're not actually endangered at all. They're fine. Emperor penguins probably are the safest of all because there isn't that much plastic down there. So emperor penguins, of course, the biggest of them, they're maybe waist high on me. I'm 6 feet, so they're about half as tall as me.
They're huge.
They're big.
Wow.
They're big, tall birds. They're 3 feet tall. And they're like this big around, but they're mostly just a bunch of feathers. They're actually not that heavy. But they're powerful. Their beaks are, yay long, but they're totally non-hostile. They're actually really curious, and they have no land predators. So we were rumbling along. So when you're on the ice, it's actually sea ice, which is, at first it feels very uncomfortable to be driving over sea ice, knowing that if the sea ice broke, you would just die because you're over thousands of meters of water. But then you realize that the sea ice is 1.7 meters thick. And the way that you got to McMurdo was you landed on a C-17 Globemaster plane. That Globemaster plane is big enough on the inside to park 4 tanks. And this plane makes a 747 look tiny. So that's how you fly in, and that lands on the ice. So I was like, if they can land that thing, we're okay in our little vehicle. And the vehicle has tracks, not wheels, because you need traction. So you're in like this little tank with these treads, and we're just rumbling along in the back of our thing, and then it stops. And usually that means there's a crack in the ice, and they have to test to see if it's too wide to cross. The rule is it has to be less than 1/3, because you need 1/3 on either side, so you can drive across if the crack is less than 1/3 of your wheelbase. So I thought we were just going to test another crack. So I was like, all right, I'll get the pole, I'll get the ice shavings. And they were just like, penguins, get down. And it was the males that were returning from doing their brood, right? So they've now been without food for months, like all the nature videos you've seen. So they're kind of hungry, but they see us, and we're all in our big red parkas, the U.S. Antarctic Service parkas, and they just waddle over to us. I have video of it. It's funny. And they get closer, and we're just sitting, and it's like 400 meters away. They came as close to me as my hand. They were just like, what's going on? And they're all talking to each other. They're making these guttural sounds, and these, like, you've seen the videos.
Yeah.
They're talking to us. They're talking to each other. They wandered behind us. They like, one of them hopped up on the tread, was like, what's this thing? They were just totally curious. And then eventually they waddled away. Also fun is that they swim on land because they're really unwieldy. So they'll lie down on their bellies and they'll just sort of push themselves along. And then when they want to stand up, they lean forward with their beak, and they use their beak like one of those backhoes to position themselves back up. So they put their face down and then they beak themselves up. So they're all rolled over. Yeah. Cute.
They do like a backflip to stand up.
Yeah,
So cool.
they basically push themselves up from their face.
That's so cool.
Yeah, imagine using your nose to stand up.
Oh, sheesh.
I can barely do it with my arms.
Okay.
Important question. Did you see any baby penguins?
No.
No.
No, those would be at the colony. I got to hear all about them because one of my roommates was a penguin biologist who had just come back and telling me mostly about the smell, which is god awful apparently. A million penguins. So yeah, not
Lots of
great.
dead fish. Yeah, probably
Lots
not.
of dead fish, lots of poop. Yeah, it's not the most pleasant smell. I've been on bird colonies before. on Nuevo Island up north, it's awful. But yeah, they got to see the little fuzzy goslings. They're cute.
That's all I asked. Did you see the fuzzy ones?
I did not see the fuzzy ones. I'm sorry.
Shame. You saw the large ones though, and those are also adorable. So
I did see the big ones.
Awesome. Well, any other questions from either Nancy or Jen?
You can ask me about my science too. It's fine.
Don't have any questions, but that was a really great presentation that you did.
Oh,
And
thanks.
very interesting stories.
I like telling stories.
Yeah,
It's the best part of professoring.
love it.
Awesome.
Well, thanks for everybody coming in.
thanks for
coming
giving
to
this
speak to us.
presentation.
Yeah, thank you so much.
And I hope anybody who watches this at home gets enjoyment as well.
Yeah, it's gonna go on our Santa Rosa Island. I
Oh,
think
cool.
it's our YouTube channel. We have some other people that definitely watch it that can't make it, so it will be seen.
Nice.
Nice.
Well, hello to the audience at home.
Awesome. Well, I guess I'll stop the recording now then.